Good morning, student. I am Dr. Reeta Kumari, Associate Professor, D.A.V. PG College, Siwan. My topic is Enzymes for TDC Part 1 students. Enzymes. Enzymes are protein that function as biological catalyst. A catalyst is a substance that speeds up a chemical reaction but isn't changed by the reaction. Now, enzymes catalyzes all aspects of cell metabolism. Property of enzymes. Enzymes are highly specific to the reaction they catalyze. That is, the specific enzymes can perform a specific function. Oxidase enzymes only help in the oxidation reduction reactions like 1. They alter or speed up the rate of chemical reactions that occur in a shell. They remain unchanged after the chemical reaction. They are affected by temperature, affected by pH, and they catalyze reversible reactions also. Distribution of enzymes. Enzymes are not randomly distributed but are specifically located inside the cell. For example, digestive enzymes. Digestive enzymes present in the digestive gland like pancreas and the stomach, which is trypsin, chymotrypsin, and the pepsin, which is present in the stomach. Glycosol, which help in the oxidation of glucose, and in the mitochondria, all the enzymes of the TCA cycles are present. Now, classification of enzymes. Enzymes are classified into different classes. Now, class 1, oxidoreductase. Oxidoreductase are those enzymes which help in the transfer of hydrogen or addition of oxygen. Example, lactate dehydrogenase or succinate dehydrogenase. Class 2, transferase. Transferase enzymes help in the transfer of group other than hydrogen. Example, aminotransferase and hexokinase. Hydrolase, it cleave bond and add water. Example is trypsin. Class 4, lyase, it cleave bond without adding water. Example, aldolase. Aldolase enzymes which help in the cleaving bond. The best example of aldolase is seen in the fourth step of glycolysis cycle. Class 5, isomerase. It helps in the intramolecular transfer. Example, triose phosphate isomerase. Class 6, ligase. It is a ATP dependent condensation of two molecules. Example is glutamate synthetase. Now, composition of enzymes. Enzymes consist of apoenzymes. Apoenzyme is a protein portion consisting of amino acid chain. Second, cofactors which activate the enzymes and are not protein part. And the holoenzymes, which is a conjugated enzymes. Both cofactors and apoenzymes help to form a conjugated enzymes called holoenzymes. Now, substrate and the active site. Enzymes operate by binding of a substrate, and the second is has the catalytic active sites, the portion of enzymes where substrate binding occurs. Now, factors affecting the enzymes activities. There are several factors which help in the affecting enzymes action. These are enzymes concentration, substrate concentration, product concentration, temperature, hydrogen ion concentration, presence of activators, presence of inhibitors, presence of repressor, and the covalent modification. Number 1, enzymes concentration. How the concentration of enzymes help in the enzymes action? As the enzymes concentration increase, the rate of reaction is increased linearly. At very high enzymes concentration, the substrate concentration may become rate limiting, as shown in the graph. Second, substrate concentration. As substrate concentration is increased, the velocity is also increased in the initial phase. As more substrate is added, all enzymes molecules are saturated, and further increase in the substrate cannot make any effect in reaction. The maximum velocity is obtained at Vmax. Temperature. Enzymes have an optimum temperature at which they work fastest. Up to the optimum temperature, the rate increase, and above the optimum temperature, the rate decrease, as shown in the figure. Now pH. Enzymes have an optimum pH at which they work fastest. For most enzymes, this is about pH 7 to 8. But a few enzymes can work extremely pH, such as protease enzymes in the animal stomach, which have an optimum pH 1, as shown in the figure. The pepsine better act at the pH 1.5 to 2. Amylase better function at 6.5 and arginase better function at 10.5. Role of enzymes in human body. There are different roles of the enzymes in the human body. It helps in the degradation reactions, which is also known as the catabolic reaction. Second, it helps in the synthesis, known as anabolic reaction. It helps in the digestion and also it helps in the protection. Enzymes are also used in the protection against the pathogen. They can be used to destroy invading microorganisms. Now, what are the applications of enzymes? There are several applications of enzymes and few are: number 1, in medical application, L-asparaginase remove L-asparagine from the tissue needed for the tumor growth. It used in the treatment of cancer in leukemia. Urokinase, it break down the blood clot and used in the treatment of heart disease. Analytic application, glucose oxidase, it oxidizes glucose and used to test the blood glucose. Luciferase produces light and used as the chemical indicator. Industrial application, alpha amylase help in the breakdown of starch and used in the food industries. Renin help in the clotting milk protein and used in the cheese making. Protease help in the digestion of protein and used in the washing powder. Now manipulative application, lysozymes, it break 1,4 bond and used in the destruction of bacterial cell wall. And at last endonuclease, which break DNA into fragments and used in the genetic manipulation on DNA fingerprinting. Thank you.